So in comparison to carbohydrates, lipids have less oxygen. They have a greater than 2-to-1 ratio of hydrogen to carbon. And so it's all, in carbohydrates, it's always 2-to-1. It's exactly equal to 2-to-1. So this is where they are greater than 2-to-1. And so you're going to have like huge long chains where you have carbon, 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 and you just have hydrogen, 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 and more hydrogen, hydrogen, hydrogen. So lots and lots of hydrogen repeating all the way up there. And then you might at the end have like a carboxyl group or something like that. Or a methane group or something, methyl group. But this is a pretty common, like fatty acid chains. They're going to be much bigger than this, but they have an acid group at the end and lots of hydrogens. So where in a carbohydrate you might have OH on one side and H on the other side, every carbon has an OH and an H. These just have hydrogens, and so there's very few oxygens. And oxygen, we said, was one of those electron hogs, and so it makes it really polar. And so this is one of the characteristics, the fact that you have less hydrogen or less oxygen means that it's less polar. In fact, most lipids are non-polar. And so fatty acids, glycerol, these are not going to mix in with water. And those are the monomers. There are other monomers, like you could have an OH group, that's an alcohol. But generally there's some sort of fatty acids involved and then some other, but there's not just like one set of monomers like we have with carbohydrates. It's monosaccharide, and with proteins it's amino acids, but lipids, they have lots of different examples. There's no set name for the dimer, and like our polymers, we have things like triglycerides and phospholipids, cholesterol, sterols. So those would be a few examples. Triglycerides have stored energy, and so it's very energy dense and lots of energy there. Phospholipids are in the membrane, and then cholesterol is in the membrane as well, but cholesterol is going to give kind of stability to the membrane, and steroids are going to be like a hormone. So they're going to regulate. They're going to have a regulatory function. So the operational term for substances that are not soluble in polar substances such as water, but are soluble in nonpolar substances such as benzene and chloroform, is the definition of a lipid, but they have just long, complex hydrocarbon chains that are hydrophobic, so very largely hydrophobic. Triglycerides are an example. They're for storage of lipids, fats, and oils, and they have a 3-carbon glycerol and 3 fatty acid chains attached, one to each of these carbon chains, to make a triglyceride. And they use also dehydration synthesis to form ester bonds. And so you have glycerol, and you take a hydrogen from one and a hydroxyl from the other, and so each of these forms a ester bond in the middle. So you have a C double bonded to O and an O, COO, and then the fatty acid chain goes in and is attached to it by this ester bond. So fatty acid chains, you can see the acid group over here, the carboxyl group, and then the ester is the bond, and you oftentimes will have this CH3, which is a methyl group, which probably is not that big a deal. I don't think I really ask you a methyl group as one of the functional groups, but that's at one end and the carboxyl is at the other end, and then it's just all these carbons with hydrogens. And it's called saturated if there are no double bonds and it's completely full. So no double bonds. and it's completely full of hydrogens. So here we have some double bonds, so this is considered unsaturated, and there's three double bonds, so we call that polyunsaturated. You'll notice the difference in the shape here. These are very straight because of the lack of double bonds, and these are kind of kinky, and that's not really a great word, but it is what it is. So it bends. There's lots of bends in these. Wherever there's a double bond, there's a bend in that chain. So fatty acids, saturated, typically saturated fatty acids are going to be found in like animal fats, but they have single bonded, and all of the carbons have two hydrogens attached to them. So they're full of hydrogens, and they are going to be solid at room temperature. So this would be like lard would be an example. Unsaturated fatty acid, generally from plants, and these are usually oils, so they're liquid. They're a liquid at room temperature. And they have at least one double bond that exists between the carbons, and that's what makes them unsaturated. So they could potentially react with and make a hydrogen bond there, but they don't have it. And so you'll see again they have a bend in their chains here whenever there's a hydrogen or a double bond, and they are pretty straight. And I say straight, but in actuality, if you look at them, it's kind of a zigzag back and forth, and that's why they show it like a zigzag here on those nonpolar chains. So a phospholipid has an alcohol group, a glycerol, and the glycerol actually is bound to the two fatty acids that make up that chain. So it's phosphate, a variable alcohol group, and then your glycerol. So this is very polar up here. This phosphate head, the head is what they call that, head. The head is polar, and the tail section is non-polar. So this section is hydrophilic, and the tail section is hydrophobic, and it allows us to make a phospholipid bilayer, which is what the membranes of the cell membrane, and other membrane-bound organelles are going to consist of phospholipids. And other things will be dissolved in these, but that's what makes it so that it's like you've got a hydrophobic layer where the fatty acids are touching, and then the hydrophilic layer on the outside where the water is. So this is going to be something we'll talk about more with the membrane, but you've got different molecules, like the cholesterol is embedded in the hydrophobic layer, and phospholipids make up the layer, and then the hydrophobic section of a globular protein or one of these transmembrane proteins is embedded into the hydrophobic layer of the membrane. And so that kind of helps to situate the proteins in the membrane properly. So the hydrophobic face is outside and inside of the cell, and the tails are kind of in the middle of the membrane. So it's sort of like a hydrophobic sandwich here. So the two pieces of bread are the phosphate heads, the hydrophilic area, and the hydrophobic area is in the middle. So steroids, they're ring structures. They're commonly found in the membranes as cholesterol, and that reinforces the cell membrane, makes it a little bit more stable. And you see this in cells, bacteria cells that do not have a cell wall, like the mycoplasma bacteria. Ergosterol is another steroid that's in the cell membrane of fungi. Wax is basically something that algae and plant cells, it gives kind of a waterproofing thing, and it's an ester formed between alcohol and fatty acids, and it's soft and pliable when it's warmed, and it's water resistant when cold. And we see this in animals as waterproofing in skin, fur, and feathers. Bacteria has a really waxy mycolic acid. The tuberculosis is an acid-fast bacteria. Leprosy is also caused by a bacteria that has this mycolic acid in it, and it repels the stains, and it makes it so that once you do get it in there, if you were able to get the stain in there, then it's not going to be able to decolorize afterwards. So that's some application to our microbiology, our microbes.